All right, ladies and gentlemen, so today we are going to wrap up discussing how the actual cell division process occurs. So up to this point, we've talked about the basics of how cells go through mitosis. Okay, so now we've divided the nucleus, but we still need to divide the cell. And so that's where cytokinesis is going to come from. So when we look at cytokinesis, the actual division of the cytoplasm, this does happen a little bit differently depending on whether you're talking about it occurring in an animal cell versus a plant cell, because remember that the plant cells have that cell wall, which creates some additional rigidity in the cell, and so that's going to force us to need to divide that cytoplasm a little differently than we are capable of doing in the more pliable animal cell. So when we look at animal cells, they are going to divide their cytoplasm via these contractile rings of microtubules that form here in the center of that cell, kind of around its waist, and literally sort of like pulling string to cut them in half. So they form this cleavage furrow, and as that ring of microtubules continues to contract, it gets smaller and smaller. We eventually divide them into two daughter cells, kind of like I said, pulling apart maybe two balls of dough or something like that. Your plant cells, on the other hand, can't do that. That original cell wall that was in place is pretty rigid. So what your cells will do instead is, as they sort of elongate and push the cell as much as they can, they do create a plate here, or a space, I should say, here in the middle, where we'll get these little vesicles that'll form and line up and eventually fuse together. Those vesicles are full of cellulose, which is one of the major um, compounds that we find in your cell wall. As that cell plate fuses then with the original cell wall of the plant, the parent cell, it will eventually mature as we add some additional components and additional layers to that to make that new cell wall and finish the splitting of that cytoplasm to create those two daughter cells. So when we sort of look back on this, I showed you the slides from the lung cell of a newt. We went through the initial stages of mitosis. This is just another way to look at it, but now we're looking at it in an onion root tip. And this is a pretty classic set of images, classic set of slides that most students come across at some point throughout their schooling to look and identify the different stages of mitosis. It's a really good classic example because we're looking at the tip of an onion root. So it's a actively dividing region. It's easily accessible and stains really well. So we can see all of these different components here in the nuclei as they split apart. And then here you can see that cell plate starting to form as this cell is not only finishing telophase, but starting to go through cytokinesis as well. Okay, so we've spent all of this time talking about cell division in eukaryotes. Yet, we are not the only cells that also need to divide. Your prokaryotes also need to go through this process as well, but keep in mind they're not going to go through mitosis because mitosis is the division of a nucleus. Prokaryotes don't have that. So their process of cell division is going to look a little bit different. And so when we talk about prokaryotic cell division, they're going to do this by the process called binary fission. And really, it's just very simplistic mitosis. I don't have spindle fibers, I don't have centrioles, I don't have a nuclear structure to get out of the way. And so really, binary fission is just a matter of the bacterial chromosome replicating, and as it replicates, it pushes against the opposite sides of the cell, stretches out the cell, and then cytokinesis does come in and ultimately split the cell into two pieces. Um, Now, when we look at the replication of that bacterial chromosome, it is going to have to start at what we call this origin of replication. So keep in mind that your prokaryotic cell's genetic information, they have a single chromosome as opposed to multicellular chromosomes that we typically see in eukaryotic cells. The other thing is that that prokaryotic chromosome is also circular, so it loops back on itself rather than the linear chromosomes that we see in eukaryotic cells. So that means that that origin of replication is going to have to open, replicate in a loop, and then complete itself. So as we create those origins are what ends up pushing out to either end of the cell as we complete the replication of the rest of the chromosome and get two copies of it. Those will then end up on either end of the pole and then we'll split from there. So we see this really simplistic version of cell division in prokaryotes, but a relatively complex one in eukaryotic cells. So how did that process of mitosis evolve? Well, it's actually kind of interesting because we know that prokaryotes preceded the evolution of eukaryotes by probably billions of years. And so that means that that process of mitosis would have likely evolved from that original binary fission process. So when we look through living organisms that we see in life today, we actually see some slight variations on that mitotic process that look somewhere like an intermediary between that binary fission and the mitosis we talk about now in eukaryotes. So when we look at things like dinoflagellates or diatoms, we actually see some of the structures that we see in mitosis, things like microtubules and chromosome division happening, but they actually end up happening within their nuclear envelope. So although we talked about that endosymbiotic theory and the mitochondria and the chloroplast being major organelles to support that theory of how prokaryotes ate other prokaryotes became eukaryotic cells based on that symbiotic relationship that they formed, we also think that a lot of other organelles, including your nucleus, may have come from that same process. So it's interesting to see how when we look at things like dinoflagellates and diatoms, where their cell division process, their genetic division process, actually happens within an intact envelope, much like what we see with prokaryotic binary fission. Um, and so we can kind of see some of the evolution of that mitosis process in looking through some of the sort of lower order, less complex organisms that exist that presumably would look a little bit more closely like ancient eukaryotic cells. So that's pretty cool.